This is The Cell Cycle by Emily Simone and Izzy Connolly, in chalk. Mitosis is a division of somatic non-sexual cells. A defining characteristic of mitosis is that the daughter cells are exact replicas of the parent cell.
The cell cycle is the life of the cell from the time it is first formed from a dividing parent cell until its own division into two cells. The cell cycle is divided into four parts. Interphase, which is composed of G1, S, G2 phases, and the mitotic M phase. Like we said earlier, interphase is composed of three phases, G1, S, and G2. During all three phases, the cell grows by producing proteins and cytoplasmic organelles. Only in S phase, however, will the cell continue to grow while copying its chromosomes. The S phase occupies about half of the cell cycle.
Interphase is then followed by prophase. In prophase, the chromatin coil and condense into chromosomes, shown in pink. Chromatids then form when the chromosomes join together at their centromeres. As this is occurring, the nucleoli begins to disappear. Lastly, the mitotic spindle, shown by the green lines, begins to form and propel the centrosomes away from each other, which are shown in orange.
Next is the longest stage of mitosis, metaphase. Metaphase begins with the centrosomes at opposite poles of the cell and the resulting formation of the metaphase plate, shown by the dotted yellow line. The chromosomes then align themselves so that their centromeres lie on the metaphase plate. On each side, mitotic spindles attach to each sister chromatid through their kinetochore.
After metaphase begins anaphase, the shortest stage of mitosis. Anaphase begins by the separation of the sister chromatids, which are still aligned along the metaphase plate. This separation begins with the cleaving of the cohesion proteins. Next, each separated daughter chromosome starts to move to the opposite poles of the cell as their microtubules shorten. By the end of this stage, each side of the cell has equal number of chromosomes which are also genetically identical.
Finally, we've reached the last stages of mitosis, telophase and cytokinesis. During telophase, two daughter nuclei form at opposite sides of the cell. The nuclear envelopes consist of fragments of the parent cell's nuclear envelope, along with other parts of their endomembrane system. The division of one nucleus into two identical nuclei is now complete. In cytokinesis, a cleavage furrow occurs, shown by the pitching of the cell, and the cytoplasm divides into two. At the end of cytokinesis, two separate daughter cells have formed and mitosis is complete.
The other type of cell division is meiosis, which is composed of two parts, meiosis I and meiosis II. Since meiosis ultimately results in four daughter cells, meiosis I is the initial division and meiosis II is a secondary division of those two daughter cells. The main characteristic, however, is that the daughter cells have an original unique genetic makeup, which is why meiosis only occurs in germ or sex cells. Meiosis I begins when the chromosomes condense and crossing over occurs. Crossing over is the exchange of corresponding DNA segments in non-sister chromatids. This allows for the daughter cells to have an original DNA makeup. By the end of prophase I, each homologous pair has one or more chiasmata, which is a point where crossing over has occurred. In late prophase I, the microtubules begin to attach to the centromeres of the homologs. In metaphase I, the homologous chromosomes are now aligned on the metaphase plate. One spindle fiber is attached to one homolog, while the other spindle fiber is attached to the sister homolog pair. Similar to mitosis, in anaphase I, the breakdown of proteins allows the spindle fibers to pull apart the homologs, therefore separating them. These homologs then move towards opposite poles pulled by the spindle fibers. Telophase I begins with each half of the cell containing a complete haploid set of chromosomes, each with at least one chiasmata. Cytokinesis then begins to occur with the cleavage furrow forming in animal cells and a new cell plate forming in plant cells.
Meiosis II, or the secondary division of meiosis, begins with prophase II. Spindle fibers form once again simultaneously in each new daughter cell. Next, in metaphase II, the chromosomes, now composed of two sister chromatids, again align themselves along the metaphase plate by their centromeres. The spindle fibers attach themselves to the chromosomes by their kinetochores. Following prophase II, anaphase II is where the proteins holding the sister chromatids together break down and the chromatids separate. The spindle fibers pull the chromatids apart and pull them to opposite poles of the cell as individual chromosomes. Finally, in telophase II and cytokinesis, meiosis comes to an end. The nuclei form and cytokinesis divides the cytoplasm. The meiotic division of one cell produces four daughter cells, each genetically distinct and each with a set of haploid chromosomes.
So, in closing, a few points to remember. Mitosis only occurs in non-sexual cells called somatic cells. This is because the daughter cells are exactly genetically identical. Meiosis only occurs in sex or germ cells. Each cycle of meiosis results in four genetically unique daughter cells. Also, remember, as long as there is life, there is a never-ending cell cycle. Thanks for watching our video and have a good day!